So now we are going to observe or try to understand the organizational levels of an organism. So to do this, we have an activity where we observe a sample of, I have a pond water here, or you can use the water in which you have soaked hay for some 2 or 3 days. From that hay extract also you can observe. So here I have pond water. From this I am going to prepare a temporary slide and observe to see the microorganisms present. So those are the unicellular organisms made up of just one cell. At the same time, I have here the permanent slides. Here also I can show you all the unicellular organisms as well as how cells are arranged in a multicellular organism. So I will start off by making the temporary slide. For that, as you all know, we need a very clean glass slide, a paintbrush to transfer the solution. So here it is pond water. So I take 1 or 2 drops of pond water, and this has to be covered with a cover slip. And when we cover it with a cover slip, we have to make sure that air bubbles don't get trapped inside. So to do that, you all know the method. Keep it slant and place it on the sample. So now we have to make sure the microscope is on. Everything is known to you, and it has to be in low power, and there should be enough light falling on the stage. Then we place the slide that I prepared on the stage, and I'll make sure that the specimen is in place with the lens. Right. So now we will observe it under low power. So here when I observe, I can see some microorganisms. You can see unicellular structures. So students, in a water sample like this, you can observe usually the amoeba or paramecium. Those are the unicellular organisms or microorganisms that are usually present in pond water. So in this also we can observe the unicellular organization of organisms. So after that, I will remove this slide. This is the temporary slide I prepared. Then here I have some permanent slides. Here again you can see I have the paramecium here. You can see the slide. That is a permanent slide of paramecium. So if I observe that under light microscope, again to set it, to make sure that the sample is in position. Yes. So if I make the adjustment, I can see the paramecium. So can you all see the unicellular structure of paramecium, students? It's under low power, so that can be observed clearly. Then the same way, I have already selected some unicellular organisms. There is amoeba here. That is again a permanent slide of amoeba. So that also can be observed under microscope. Now this you all have experience observing it before also. Okay. So can you all see that clearly, students? That is again a unicellular organization. Just one cell, that is a living organism. So we are discussing the organizational levels of living organisms. Same as that, I have an euglena here, a permanent slide of an euglena. That also can be observed clearly. So here again, can you all see the cell of the under low power itself? You can see that unicellular structure of euglena. Okay. So students, I have another slide, that is Chlamydomonas. That is also a unicellular organism. So if I place the permanent slide here, I am sure you all have observed this before also in your previous grades. But again, when we observe through the low power, you can see the unicellular structure, the green color, cells, unicellular organism, chlamydomonas. Can you all see it clearly? Yes, it's very clearly visible. So these are all the unicellular organisms. Now this is the permanent slide of a heart. You all can see that clearly. So in this, what happens? The cells combine and form the tissue, and the tissues combine to form the organ. So I will observe this under the microscope. Again here, first under low power. So when we observe, now can you all see those cell structures? This is the tissue present in heart. The tissue present in heart. Is it clear to you all, students? So this is part of a multicellular organisms. So can you all understand the relationship? There are organisms that are only unicellular. At the same time, there are organisms where you have the organizational levels, and this is the organizational level where you see the cells and the tissues. You can't see the cells individually, but you can observe the tissue of the heart. So now we are going to do this activity to observe that carbon dioxide is released during respiration. To do the respiration, I have a frog here. So you can see it's a live frog. So I will have to transfer the frog into this bottle just before I start the activity, because there are holes here, so the frog will be fine and alive. So you all know, students, here we have 5 bottles including this large bottle. First one, bottle A, contains potassium hydroxide. You all know the purpose. It is to absorb carbon dioxide that is present in atmospheric air. Then the second bottle B contains lime water, that is calcium hydroxide, that is to test whether there is any carbon dioxide in air that enters from bottle A into bottle B. Then we have bottle C, where we put or keep the frog. Instead of the frog, you all know what we can replace it with. We can use a small rat, or we can use germinating green gram seeds. So those are the organisms that will respire. Again, inside bottle D, we have calcium hydroxide, that's lime water. So in 2 places, this is to confirm, bottle B is to confirm that there is no carbon dioxide in the air, and then the bottle D is to confirm that carbon dioxide was released during the respiration of frog. And then we have bottle E, that comes with a tap. So, and it's filled with water. When I open the tap, you can see the water level will decrease. Because of that, there will be a space created, a vacuum. In order to fill up the space, air will flow from bottle D to this, C to D, B to C, and therefore from atmosphere, from bottle A up to bottle E, there will be a continuous air flow. And from that, the frog will respire. So it will intake oxygen and release carbon dioxide. That is what we are going to test now. So you all have to observe these 2 bottles carefully. Now you all can see both of them, they are colorless solutions. Lime water in bottle B and lime water in bottle D, both are colorless solutions. Once we have the frog here, you will see a difference. That is what you have to observe clearly. Okay, students. So now I have the frog also in place. You all know why we are having all these bottles. Now I am going to open the tap so that the water will release, and you can see, you all can see the air flow, the bubbling of the gas. Observe bottles B and D carefully. Bottles B and D carefully. You also can see the frog breathing. When I open the tap only, you all see the flow of air. When we close the tap, there is no air flow. Here you can see, students, there is no color change. Here you might observe a slight color change. You know what the color change is. Clear solution to a milky solution. So now here, students, you all can observe the color change. Look at this bottle. The color is still the same. It's a colorless solution. Look at this bottle carefully. You can see the solution has turned milky in color. So this is the activity. Initially when there was air here, carbon dioxide was absorbed in this bottle by KOH. So therefore there is no color change with calcium hydroxide, that is lime water, in bottle B. But then the air comes into this bottle, so the frog respires, releases carbon dioxide, and that carbon dioxide gives a color with bottle D, the calcium hydroxide, that is lime water, present in bottle D. So that is the observation that confirms that carbon dioxide is released during respiration. So now we are going to do this activity to demonstrate that oxygen is inhaled during respiration. So to do that, I have two conical flasks, similar conical flasks. Inside that, we have the same amount of germinating green gram seeds. So you all can see the sprouts coming out in both of the containers. And here I have the cork with two tubes, glass tubes inserted. One is connected with a rubber tube. Now the purpose of this rubber tube is that when I fix this to the conical flask, so what will happen is, now before I fix, I will show you all, it's only the two tubes that are there. There is no other container there. So when I fix it like this, there is lot of air inside. The air can escape through this rubber tube, and once we fix it and make it airtight here at this point, we have to fold this tube, and we can either tie it very tightly with a thread, or else we can put this crocodile clip so that it is airtight. At the same time, I have the other setup, same thing, but here you can see, students, inside this small test tube, I have taken a little volume of potassium hydroxide. You all know the purpose of using potassium hydroxide. What is it? Potassium hydroxide absorbs KOH. So that additional test tube, small test tube with KOH is there in this setup. The same purpose, the glass tube connected with rubber tube, so that when we fix it tightly here, air can flow out. So the pressure will be balanced inside both of the conical flasks. And after that, we will have to fold this and tighten it, so that after this point, when the setup is going to be observed, there is no air flow inside. So now you can see I have the colored water in both the beakers. That is to make the observation clear. When the liquid, the water, rises up in one of the tubes, you can see it very clearly. So this is the setup. Now we will observe it. So now observe both the tubes. Here also you will see a slight rise, but it will not rise any further. That is due to the nature of water. It rises up a little bit into the glass tube. In both, now you can see the water levels inside the glass tubes are almost the same. Observe the setup carefully. So here you know, students, in this we get the carbon dioxide absorbed by potassium hydroxide, and we assume that the amount of carbon dioxide inside the conical flask, the air that is inside the conical flask has carbon dioxide, but that volume is negligible. And also the volume of oxygen inhaled is equal to the volume of carbon dioxide, that is exhaled. So because of that only, you will see the observation. So now here you all can see the water level rising in this setup. So that is because oxygen is being inhaled, carbon dioxide is evolved, but that carbon dioxide is absorbed by KOH. So to fill that space, air from the glass tube flows into the conical flask, and to fill that space, water from the beaker rises up the glass tube. But that does not happen in the second setup. Why? Because there is no KOH. The volume of oxygen that is inhaled is equal to the volume of carbon dioxide that is exhaled. So there is no space available for the colored water to rise up. So here now you can see, students, as I speak even, you can see the rising up of the colored water. So this observation is very clearly visible to you all. So you can see now the colored water level has risen up to somewhere here. So from this activity, what can you all conclude? We can conclude that oxygen is used for respiration, or oxygen is inhaled during respiration. So you all can clearly see the observations, the difference in the two setups. So now I am going to demonstrate how to set up an oxynometer. You all know the purpose of using an oxynometer. We use it to observe the growth of plants. And here there is an advantage. Even when there is a small growth in the plant, small increase in the height of the plant, you can see a larger change in the indicator. So because of that, in the scale you can see a larger deviation. Because of that, it's very easy to observe the growth of plants. So here these are the things that we need. We need a potted plant which is used to observe the growth, and here you need a stand. We have used a burette stand there that you can get from the lab, and here the scale, it is a wooden scale that is fixed here, and the wooden scale has this plank. Normally you are supposed to connect this pulley to a higher support so that it doesn't move downwards, but here since we have this plank, the pulley is attached here, and to the pulley you can see the indicator attached to it. So then what do? We need to attach the potted plant. Now can you all observe that? Where do you tie the thread? The thread has to be tied to the shoot apex where I have tied to the plant. You can observe that clearly. This is the shoot apex of the plant. You have to tie one end of the thread to the shoot apex. You have to send the thread around the pulley, and on the other end of the thread we have to hang a small weight. Now here this is a cork, so that's not very heavy, it's a light thing, so that the plant will be erect. So what will happen now? The plant will grow, the height will increase. Because of that, the thread will move down. Because of the weight, it will be always straight, so the thread will move down. With that, the pulley will rotate. And when the pulley rotates, the arrow attached to the pulley will move. And that is the indicator or the pointer here. You can say it's a pointer there, and there is a red colored arrowhead there, so that shows the scale. Now it is at this 90 degrees point, you all can see. So when the plant grows, it will increase, it will move upwards. So you can see the change. Now here you can see I can move it. So like that you can observe the change. So as I said before, students, the advantage of this is when there is a small growth, the pointer shows a larger deviation. So it's very easy to observe the growth throughout a period of time. So that is the function of the oxynometer. The points you have to remember are that how you attach the thread to the plant, and also the reason for hanging the small weight and why it is advantageous. So if you understand all this, you can also make an oxynometer at home and observe the growth of a plant.